The third video lecture is picking up here, looking at this graph, comparing passive immunotherapy to active immunization. In this graph, we're looking at the changes in antibody concentrations over time. As you remember, passive immunotherapy is when an individual receives antibodies, whether it's to protect against a toxin, a venom, or perhaps even an infection. In this scenario, you're giving the individual antibodies, and as you would expect, the antibody concentration starts out high, as it does, but as time progresses, that antibody is going to be degraded and the concentration is going to decrease over time. Contrast that with active immunization, where an individual is exposed to the antigen and allowed to stimulate their own immune system to generate antibodies. In this case, you're going to see an increase in antibody concentration over time. Also on this graph, you can see that booster doses were administered here and here, and that resulted in spikes on the graph because they increased the concentration of circulating antibodies. That's the comparison of a visualization between passive immunization and active inoculation. We're going to look now at some testing techniques in which the antibodies in the blood are used to do diagnostics. This is called serology. Serology is a study and use of antibodies in the blood to look at antigens or the concentration of antibodies to those antigens. There are tests that are direct testing, which use specific antibodies to look for antigens. Then there's indirect testing, which is looking for antibodies that have formed against antigens. Depending on what you're trying to do or what your suspected diagnosis is, you will choose a test. Other factors to consider are things like cost. Some tests cost a lot more than others. And time. How fast or how urgently do you need the results? All of these things are going to dictate what kind of test you might want to do. One of the simplest types of tests is a precipitation test. You mixed antigen with the person's serum, which has antibodies in it, and you're going to look for signs of precipitation. However, this is not done blindly. You have to test dilutions of your antigen or your antibody to get the optimum antibody-antigen concentration. What am I talking about? As shown in this bottom panel, in order to get a precipitate, which is a solid coming out of solution, you have to get optimal proportions of antigen and antibodies. When you reach that point, you get these complexes, as you see right there. One antibody is bound to multiple antigens that form a network or cross-linking and result in a larger particle that comes out of solution. These particles settle out and are visible at the bottom of a well or a tube. When you have too much antibody, which is shown here at the left, you end up with antibody bound to multiple antigens, but the cross-linking that's necessary to get a large particle to come out of solution doesn't form. So in these precipitation reactions, you're going to test a dilution of antigen and antibody over a series of antigen-antibody concentrations to see if you can get these optimal proportions that can result in a precipitate. When and if it does, this tells you that yes, you do have the right antibodies present in the test serum. It's a fairly simple and inexpensive test, but it's not the most sophisticated, and there are other ones that would probably be used for diagnostics. Another low-tech precipitation test is a diffusion test. Here, however, you're using a gel, something like the agar gels that you see in a petri dish, but without the media or nutrient components. In these diffusion gels, you have two wells punched out of the media. In one well, you place the antigen that you're interested in, and in the other well, you're going to put your test serum with the antibodies. You allow them to diffuse towards each other, and again, this is a precipitation reaction. So you're looking for this visualization, a line of precipitate that comes out when the optimal concentration occurs. Why? When you're diffusing, you're automatically getting a gradient of antigen and antibodies. And at some point, you hit the optimum precipitation zone. And that spot will result in a precipitation line, which tells you, yes, you do have the antigen of interest. If you have multiple antigens that might be coming from the same particular pathogen and a serum with multiple antibodies to those antigens, you would see multiple lines forming when you get to the optimum concentration for each one of those antigen antibody complexes. An agglutination test compared to the previous tests is used quite often. An agglutination test is what's used, for example, to do blood typing tests. In this case, we're using cells that you want to cross-link and form agglutination, something much larger than a precipitate because cells are much larger than particles with cross-linked molecules. What happens during agglutination is that you mix an antigen containing substances, so in this case, our red blood cells, with antiserum, which contains the antibodies. The antiserum is going to be added to the wells, and as you can see, we have antiserum against the A, the B, and then the Rh factor. That's on red blood cells. You mix the person's blood, and you can see that you have agglutination or clumping that has occurred with the Rh factor and with the B antigen, but not with A. So this individual would be B positive blood type. The Rh is positive or negative, and then of course we have the B as the antigen. This is easy to perform and see results, and is something that's done quite often. If we're talking specifically about red blood cells, the term hemagglutination is used because we are agglutinating red blood cells, but it is just an agglutination reaction. This blood typing agglutination reaction would be hemagglutination, but we can do it with other antigens as well. This image is showing a titer. A titer is when you're trying to determine the highest dilution that you can use and still get a reaction. In this example, we're mixing dilutions of serum and an antigen, so serum is diluted, but the antigen is the same in each well. Then you look for that precipitation or agglutination reaction to occur. And you can see that it has occurred in this well, which is 1 to 1,000, but not the next well, which is 1 to 10,000. So the titer is 1 to 1,000 in this particular test. This type of test is used to determine whether you have a reliable response to a particular vaccine. For example, if you're in healthcare, you're required to get the hepatitis B vaccine. If you have had the vaccine, they'll do a titer to make sure that you still have a high enough response that's protective. If you do not achieve the titer that is needed, you do not have a high enough dilution and still see the agglutination reaction, they'll recommend getting a booster to be protected against the infection. That's a titer, and they use it to determine if you have enough of a particular antibody to achieve the protective response that you want. A neutralization test is something that you have heard a lot about in the news if you've been keeping up with the COVID-19 vaccine information, because what they're trying to do is make sure that the response an individual is making is actually protective against the virus. They're also determining if the vaccine response is protective against the new variants that have emerged. So how is this being done? To do a viral neutralization check, You are looking for the killing effect of the virus on cells. That's called a cytopathic effect. Using the correct type of human cell lines, you introduce the virus to them. Normally, the virus will kill the cells. What you're going to do then is to attempt to neutralize the virus when you mix a patient's antibodies with it and test for a decrease or absence of the cytopathic effects. Here is a tube and a cell. In the tube, you'll have virus. And prior to applying it to the host cell, a person's serum is added to the mix with antibodies. As a control, of course, you have a tube with the virus without patient serum. What you're hoping to see is that the virus is neutralized because antibodies bind to the surface, as shown there. A viral particle will have antibodies bound to it to neutralize it. You allow an incubation period for this antibody-virus interaction before you add it to the cells, and you look for a cytopathic effect. The control, which has no antibodies, will kill the cell. But if you have neutralizing antibodies, when you add this mix of serum plus virus, the cell should live. That means that you will not see killing in the test scenario because you have effective antibodies. Of course, if the antibodies are not protective, the test will have dead cells too. Besides testing for neutralizing antibodies, this is a test that can be used to determine if a person ever was exposed to a particular virus. If they were exposed, they would have the protective antibodies in their blood. If after some time has passed since the infection, say it's been a year since you recovered from COVID-19, and you're trying to determine if you still are protected from reinfection with the virus, you would run this test. If you still have neutralizing antibodies, the test well cells will live. If not, if your antibodies have decreased too much, the test well has dead cells. So this is the virus neutralization test, and it's very useful to determine the effectiveness of the antibodies in your blood. We can use antibodies to do different types of diagnostics as well. In this case, we're going to talk about labeled antibodies. Think back to those monoclonal antibodies that were produced in mice. They are often going to be used as labeled antibodies, or you can make the secondary antibodies and label that. So what are we talking about? The primary antibody is the antibody that in these tests are used first. This could be a patient's serum antibody and you incubate it with your antigen. In this case, it's on a slide. And then the secondary antibody is added second and is going to be directed against the first antibody. How does that happen? Remember, I told you that we can make an antibody against an animal's antibody, and the same is true for animals. An animal can make antibodies against human antibodies. So you could take an animal like a goat or a rabbit and vaccinate them with human antibodies, and they will respond and make their own antibodies. Then you can purify those antibodies and do what's called tagging, the placement of something on the antibody so that you can visualize it and detect it later. In this case, a fluorescent molecule has been added to the secondary antibody, and the antibody is against IgG. So this is anti-IgG, an animal antibody made against a human antibody. This animal antibody binds to human antibodies. You can wash off all the excess stuff and you can see where the primary antibody has landed because the secondary antibody is providing the label. You can see the green fluorescing cells because it tells you that the primary antibody bound to them and then the secondary antibody with the fluorescent molecule stuck to those. If you do it in one step, they call it direct. But if it takes these 2 steps as described here, it's indirect. The direct would have to be a labeled monoclonal antibody, and the indirect is what is shown on the panel, where you do 2 steps. The first, a patient antibody, and the second is a labeled antibody. We're going to move on to a different type of labeled antibody. This test is called an ELISA. It's super important, and it's a commonly used test. You're going to be doing some virtual labs and hopefully real labs for this. ELISA stands for enzyme linked because you're linking an antibody to an enzyme. The immuno is saying that we're talking about the immune response. And sorbent, meaning sticking. Assay is a fancy sciencey term meaning test. So this is showing you the classic type of an ELISA. And what's happening here is we're trying to detect antibodies to a specific antigen. It's fast, inexpensive. You can test a lot of samples at once, and this can be done automated, like with robots, types of machines. So you can test a lot of patients at a time. Typically, we're trying to determine whether the patient has antibodies to an antigen, meaning they've been exposed. The disease might be something like COVID-19 or HIV or various diseases. So going down these panels, the first thing that we do is in the well, you're going to put the antigen, in this case, that little pink thing. You follow that by coating the well with non-specific proteins, the green stuff. And this can be something like albumin or milk protein, something that doesn't affect the results of the test, to block any of the spots that your antigen did not stick to. This is an important test because you don't want the antibodies to non-specifically be sticking later in the assay because that'll screw up your results. Then finally, you have your primary antibody, which will come from the patient. And you see that it's bound to the antigen right there. So the person does have antibodies against that particular antigen. Then you wash out all the non-specific, unbound stuff. You come back with a secondary antibody. Something like goat anti-human or horse anti-human, rabbit anti-human. And that's going to be linked to your enzyme, that little green structure right there. And again, you wash it out, all the non-specific stuff. You're going to be able to tell if you have antibodies in the well, because in the last step, you're going to add the substrate to that particular enzyme. The substrate is going to go from colorless to a colored substance. So if you have a lot of antibody bound, it'll get dark very fast. If you have less antibody, the reaction will take longer to be seen. So when you have a specific assay and you wait a specific amount of time, you'll be able to compare relative amounts of response in people based on how dark the enzyme of the wells, how dark the substrate in the wells becomes in a specific amount of time. You can see here, this is a typical ELISA plate with some of the wells that are positive. You can see the different shades that are positive. This test that's shown here, which is called a sandwich ELISA, is a variation on the theme. And in this case, we're going to detect antigen in solution. The solution could be something like patient serum, but also something like a juice substance or a medical solution. And you're trying to detect if something that should not be there is present. What you'll do is, instead of coating the well with antigen, you coat it with antibodies specific to the antigen of interest. This might be a monoclonal antibody, for example, and the following steps will be similar as before. You block, and then you add your solution that contains the potential antigen, and if the antigen is there, it gets captured by the antibodies sticking in the well. You wash all the excess, and then you come back with a secondary antibody. This time, however, it's also directed to the antigen, and it has the enzyme linked to it. So now you have this sandwich of antibodies with the antigen in the center. Finally, you add the substrate, and it goes from colorless to colored. Colored wells indicate that the antigen was present. In sum, the sandwich ELISA is used to detect antigen in solution, where a typical classic ELISA is used to detect antibodies to a specific antigen. Another variation on the ELISA is called a Western blot. Why Western blot? It's kind of a joke because Southern was an individual that came up with a method to detect DNA. Then when they came up with a method to detect RNA, they called it a Northern. Right, yeah, science humor. And then when they came up with the method to detect protein, they called it a Western. So this is a Western blot. And what are you going to do is start out with a gel that can separate proteins. And this is different than agarose. It's usually polyacrylamide gel with wells. But you go through that process of electrophoresis, which is not shown right here, but you separate the proteins out by size, just like you do with DNA. Then you're going to set up to transfer the proteins from the gel to a filter paper. As shown here, it's a kind of an old-fashioned way. They do it with electricity nowadays, but it's the same idea. You have a filter paper sitting on top of this stack, and you want the proteins that are in the gel to get stuck to the filter paper, which is what's happening in this transfer called blotting. The transfer will result in the exact same pattern on the membrane as was in the gel. You can cut them into strips, and then you can put them in specific little container so each well contains a strip. You can incubate each strip with patient serum and do essentially an ELISA on the membrane paper. In this case, you'll end up with antibodies binding to the paper. Otherwise, it's working like the classic ELISA. For example, let's say that this is a band on the gel with a specific protein that landed during electrophoresis. If antibodies are present in the patient's serum, they will bind to it. Then you come back with the secondary that has the enzyme on it. And finally, the substrate, which will give you color. The advantage to this is that you can detect more types of antibodies. If you're looking at something that's complex, where you might need to see a pattern for antigen recognition, as opposed to a single interaction. Well, this is your test. For example, with Lyme disease, they like to do Western blots because you need a response to multiple antigens on the organism for a diagnosis of Lyme disease to be accurate. If you look at all of these lanes right here, 1, 2, 4, 5, and 6 people that were tested are reacting to a couple of things that are similar to the control. Know that the causative agent of Lyme disease is a bacteria, and thus it has some common antigens shared with other bacteria. For example, the flagella protein is going to be similar among different organisms. So you would expect people to have antibodies to flagella protein. But if you look at the positive control, somebody that for sure had Lyme disease, they're responding to multiple antigens. Based on that comparison to that patient right here, patient number 3, you can say it looks like number 3 does have Lyme disease because their pattern is a response to multiple antigens on this bacterium, similar to the positive control. The response to many more antigens than the other individuals. If you tried to put these antigens down in an ELISA well, perhaps all of these people would look positive because they're all reacting to some antigens on the bacteria. We can't know which. So this Western blot lets us look at a pattern of response to antigens in a pathogen and enable a more accurate diagnosis. Lastly, let's look at immunochromatography. A pregnancy test is a classic immunochromatography test. It's an ELISA modification that's performed on a little strip. They do this for lots of different things now, including HIV, rapid strep tests, and others. There's also a version of this test to determine whether you have IgM or IgG or both to COVID-19. So the antigen can be different things. If you look at the top, you have a strip that's labeled, and what you have in this area are antibodies to antigens. And they're labeled with a colored molecule. The label's called colloidal metal, but that's just a colored molecule. The color is too diffuse in this area to be noticeable. But later, up on the strip, you have fixed antibody to the antigen. One will be a control line and one will be the test line. Amongst these different antibodies, a plus mark on the top are the control antibodies, and they're going to be preloaded with their antigen so that they will show up when you dip this strip into a fluid and capillary action draws them and the fluid with antibodies moving up the strip. The control should always work. The control is antibody bound to a control antigen, like that. And antibodies up on the line, up here, should capture them. So the antibodies will get concentrated on these lines where there's antibodies to the antigen, and all of a sudden, you'll see colors develop. You should always see the control line showing up because that tells you that your strip is still good and still working. The test line will only show up when the test antigen was present in your solution. So for a pregnancy test, you're looking for the HCG antigen, but if it's some other type of test, you're looking for another specific antigen. The advantages to these is that they can be done very rapidly and they tend to be reliable indicators of specific antigens. All right, that's it for chapter 17. See you in the next chapter.